Excellent. Thank you. Thank you again for reminding me of that here. And let me switch back real quickly to the whiteboard. Oh, I'm going to lose all this, aren't I? That's all right. We'll do it again anyway. So thank you for reminding me of that. Thank you for catching that. I apologize for not recording. So yes, here's the game plan so that you have that. We talked about that already and what we're doing. Again, nothing new from what we did last time. So you have that information. So thank you for that. I apologize. All right. So that allows us to go back to the lecture now. So as we were saying, we've done a lot already on nervous physiology. Out here on the cell body and on the dendrites, we have talked about how we can produce a graded potential. Then at the axon hillock, we talked about how this is the location where we produce an action potential. From there, we worked our way down to the rest of the axon. And again, I'm cheating and using my square like that to do that. We talked about the propagation of an action potential. And we talked about doing it two ways. We talked about both with myelin and without myelin. So really, all we have left to talk about the physiology of a neuron is to talk about how it is going to communicate with the target cell, right? The goal of it is to talk to some target cell. And just out of curiosity, what are most target cells? What do most neurons talk to? I am speaking out loud, right? So effectors are not a bad guess, but that's not what most neurons are going to talk to. What do you think most neurons talk to? Muscle cells, again, would be an example of effectors, and those are things that we can talk to, but it's not the most common target cell. Well, okay, let's see if we can work our way through it. What is the most common functional type of neuron?
Multipolar.
That is the most common structural type. Yeah, absolutely. What's the most common functional type?
Interneurons?
Interneurons. There you go. Absolutely. So what is the job of an interneuron?
To carry information between the sensory motor in the
Right. To basically carry information between the sensory nervous system and the motor nervous system. So what do interneurons, since most of our neurons are interneurons, what is the target of most of interneurons? What do interneurons actually, what is the cell that an interneuron talks to? Nope, not the receptors. That's what the sensory cells receive their information from. An interneuron is between a sensory neuron and a motor neuron. So what do most interneurons talk to? Yeah, there you go. Other neurons. Most target cells are other neurons. So most neurons communicate to other neurons. Exactly. And of course, what are we going to call this specialized region for communication at the end of a neuron?
Axon terminal.
Okay, so the axon terminal and the synaptic end bulb talk to the target cell. And what's the generic term we use for that specialized area of communication?
The motor end plate.
Again, the synaptic end bulb and the motor end plate talk to each other. The neuromuscular junction is a specific example of one, but the generic term for them was a synapse. Synapse. All right. Again, this is the part that should be easy because this is the part we've already talked about. I know it's the Wednesday before Thanksgiving, and being here and lecture at 8 o'clock in the morning is probably the last thing most of you want to do, but we do have a lot of material to get through, so we're going to still push forward. So yeah, a synapse. So we've talked about producing graded potentials. We've talked about producing an action potential. We've talked about propagating action potentials. And now we have to talk about communication at a synapse. And the good news is we've already talked about this. We already know this. There's not going to be any really new information here. We may use a couple new names, but there's no new information. And that's the nice thing about this, right? Our neuron communicates with its target cell at a synapse. So when we talk about the two cells, we have a presynaptic cell. That is going to communicate to our target cell. And since we said our target cell was most commonly a neuron, let's go ahead and make it a neuron. So this we could call our target cell, or the other thing we could call it is the postsynaptic cell. All right, so a synapse is that specialized area of communication between the presynaptic cell and the postsynaptic cell. And believe it or not, there are actually two types of synapses. I know it doesn't seem that way because we haven't talked about it that way, but we have. We haven't used this term before, but the first type of synapse is an electrical synapse. An electrical synapse, as we know, is when two cells are connected by a gap junction. What were the proteins again that form gap junctions? Anyone remember? I know talking about that seems like a billion years ago, but this is a cumulative class. What are the proteins that form gap junctions? Anyone? Fun. Connexons. So those connexons, remember, are the proteins that form a gap junction. Tell me what you know about gap junctions. What is the advantage of a gap junction? This is stuff you guys should be able to do. Come on, guys. Gap junction. What do?
They talk to other cells. They send information.
Exactly. And not just do they send information, but they rapidly. Remember, a gap junction is basically just a fluid fill hole, a fluid fill pore between the two cells. So, for instance, if this first neuron had an action potential, we would get a huge, massive influx of sodium. And that sodium would be able to very rapidly spread right through that gap junction to the next cell, which would then get a massive influx of sodium as well. And notice in this case, I had this going from left to right. Do gap junctions only work from left to right? No, this is going to allow two-way communication. It can work the other way as well. So this is rapid communication. This allows two-way communication. And remember, like we talked about in smooth muscle, like we talked about in cardiac muscle, the advantage of these gap junctions, the advantage of an electrical synapse is that it allows the cell to work together as one. Right? These cells are hardwired together. So they work together as one unit. In fact, if you remember, the smooth muscle cells that had a lot of gap junctions, that's what we called them. We called them single unit smooth muscle because they work together as well, as one. However, remember when we talked about gap junctions, those are the kind of places we saw them. We saw them in smooth muscle. We saw them in cardiac muscle. We saw them in areas like that. Did we talk about them a lot in the nervous system? No. While there are places in the nervous system that are going to have electrical synapses and have gap junctions, there's not a lot. And if you think about it, that makes sense, right? When you're building a house, you want the house to be hardwired so that when I flip that switch, this light always goes on, and I flip it again and it goes off, and I flip it again and it goes on, and I flip it again and it goes off. I'm never going to flip that switch and have the front door open or the garbage disposal go off, right? Or the toilet's going to flush, something along those lines. We want it to be hardwired together. But we don't want our nervous system hardwired together. We want to be able to make decisions. Because if I stand here with my mighty red pen in my hand, I look you in the eyes and say, I'm going to kill you. And a bear with an axe walks in the door behind you, looks you in the eye and says, I'm going to kill you. You are getting the exact same stimulus, but are you going to respond the same way to both of those things? No, of course not. For starters, I'm on camera, right? And have a red pen, right? For the second thing, a bear holding an axe who doesn't even have an opposable thumb is truly terrifying, right? You're going to respond to those things differently. We want to be able to make decisions. We want to be able to modify our behavior. We don't want things to be hardwired. And so while electrical synapses definitely have their uses, definitely have their function, most of the synapses that we have in the nervous system, so we did that, are chemical synapses. Chemical synapses are what we saw in the neuromuscular junction. We had a synaptic end bulb. We had a motor end plate, right? We had, and they don't touch. There is a space in between the two. So our presynaptic cell has some specializations to it that we've talked about before. It is going to have vesicles, and those vesicles contain what?
Neurotransmitter?
Exactly, neurotransmitter. Absolutely, those neurotransmitters, right? And of course, we're going to need some special way to release them. So those usually involve voltage-gated calcium channels. Our postsynaptic cell has to have some specializations to it as well. The key one is it has to have some type of receptors, chemical receptors, let's say it that way, that are going to bind to that neurotransmitter that is going to be released. The neurotransmitter binds to them. And usually as a result of that, it's going to open some type of channel. So we have some type of channel, some type of receptor on that motor, on that postsynaptic cell. Let's use these. That chemical can bind to. And again, there can be other specializations as well. Like for instance, in our skeletal muscle cell, we saw that that motor end plate has those invaginations that increase the surface area. But those aren't required. What is required is some type of chemical receptor, some type of channel that neurotransmitter released from those vesicles. And let's go put a couple vesicles in this bad boy. there we go, is going to be able to release. Now, one of the problems isn't the right word, but one of the concerns with this is that it is a slower process. Rather than just having sodium spread from one cell to the next, as we know, calcium needs to enter the synaptic end bulb. That triggers all the events that cause the neurotransmitter to be exocytosed. It then has to diffuse across the synaptic cleft. It has to bind to those receptors. It has to open up those channels. Ions have to flow in. So a lot more events have to occur. So there's a bit of a delay in that process, right? It's not a massive delay, but it is a little bit slower. Also, whereas before with a gap junction, we have two-way communication. Do we have two-way communication here?
No.
No, in this case, it's just communication in one direction, from the presynaptic cell to the postsynaptic cell. So it's a slower process. It only allows for the one-way communication to be able to take place. But the advantage of this is that it is modifiable. We can, that's not how you spell modifiable. This can be modified. The effect that it has on the cell can be different. And this allows us to have different behaviors, right? You're walking out of the store carrying a big, huge bag of groceries and you drop them, right? And 3 people come out of the store and 3 people do that, and you could get 3 totally different reactions. One person could just start bawling right there on the spot. One person could just sigh and bend down and start picking them up. One person could just turn and walk away and never go back to that store ever again in their entire life, right? And depending on the day and your mood, you could respond all 3 of those ways as well. The advantage of this system is it's modifiable. And the way we modify it is going to be primarily by the types of channels that we open on this postsynaptic cell. As we know, some can make the cell more positive, some can make the cell more negative. Or the other terms we can use for this now is we can say that some are going to be excitatory and some are going to be inhibitory. And remember, most target cells are other neurons. So as we know, when we open chemical channels on a cell body or on a dendrite, we produce a graded potential. And so it can be a positive or excitatory graded potential, or it can be an inhibitory or negative graded potential. And the point of these is, depending on the size of the graded potential, the numbers of graded potentials, we may, but not always, get an action potential. It allows us to modify the behavior. And again, that's key. I mean, you think about it, we'd think, oh, well, you're not hardwired for anything, but then usually the response I get is, what about reflexes? Right? If someone hits you in the knee with a hammer, what happens? You go to the doctor's office, they sit you on the counter, they hit you in the knee, and what happens? Yeah, your leg kicks up as a result of that. Absolutely. Your leg kicks up in the air as a result of that. And they hit you again, and it happens. And hit you again, it happens. Does that mean that that reflex is hardwired? Well, what if instead, before he hits you in the knee the next time, they put a sleeping baby right next to your foot? When they hit you in the knee with a hammer, are you going to kick that baby? No. You can stop that reflex. You can modify that behavior, right? Unless you like kicking babies, then feel free. You have an excuse, right? But no, even reflexes are modifiable. The advantage of a chemical synapse is we can modify behavior. And we're going to do that by producing different types of graded potentials. All right. Now, we know how this works. We talked about this. Many of you had this as an essay question on the previous exam. So I'm not going to spend too much time going over communication at a chemical synapse because we've already done this. Really, the only thing that changes in that process at all is that instead of saying at the motor end plate, we just say on the target cell or the postsynaptic cell. And since most postsynaptic cells are neurons, then our chemically gated channels are going to be on the cell body or the dendrites of that target postsynaptic cell. So I know some of you got this as an essay question. I know not all of you got communication at the neuromuscular junction as your essay question, but it's the exact same process. You've already learned it, so you're responsible for it in this one as well. The way a chemical synapse works is the action potential travels down the synaptic, the axon to the synaptic end bulb, opens the voltage-gated calcium channels. Calcium triggers exocytosis, diffusion, binding to the chemically gated channels on the postsynaptic cell, opening anion channels, causing ions to flow in and out. And even just like we said before, if the depolarization is big enough, we get an action potential. So you know this process. I don't have to bother going through it again, but be aware that it is still possibly an essay question you could be responsible for on the exam. So I'll ask quickly, any questions on this process? Anybody who wasn't clear on it the first time so that we know and understand this for the second time? All right, excellent. So then we're kind of back at the beginning again. We were just talking before at the beginning of this about those graded potentials, and that's what we're going to be producing. Binding of the neurotransmitter to the postsynaptic cell opens ion channels, right? Causing ions to flow in and out of the cell, producing a graded potential. And as we've talked about, you can have two types of graded potentials, or postsynaptic potential because it is on the postsynaptic cell. They can be depolarizing, which would be excitatory, or they could be hyperpolarizing, which would be inhibitory. Let's see some examples. All right, here we see an example of an excitatory postsynaptic potential. Again, it sounds like a big fancy term, but really we're just adding a label to something we already understood. On a neuron, on its cell body and its dendrite, we can open ion channels. And when we open some ion channels, it will depolarize the cell. And primarily, which ion are we going to use to produce a positive or excitatory postsynaptic potential?
Sodium.
Sodium, absolutely. So it's usually sodium. Yes, in some instances we can use calcium, but remember, as we've talked about, calcium makes cells do wonky things. So we don't usually want to use calcium for this. All right. Now, notice our definition of excitatory. Excitatory means the cell is more likely to reach threshold. Not that it guarantees that it gets it there. Notice in this case, our stimulus didn't get us to threshold. It doesn't get us an action potential, but it's still considered excitatory. So excitatory just means we get closer. It makes it more likely to reach an action potential. But it doesn't guarantee that we get there. It doesn't have to reach threshold to be excitatory. And like we said, typically this is going to involve our sodium channels. Conversely, an inhibitory postsynaptic potential is one that makes the cell more negative, that hyperpolarizes the cell. This, of course, is going to make the cell less likely to reach threshold. But again, it doesn't guarantee that it doesn't get there. It just makes it less likely. And as we've talked about before, which ions, plural, are we going to be most likely to use for an inhibitory postsynaptic potential?
Sodium and potassium and chloride.
Excellent, careful. Chloride and which one?
Potassium.
Potassium, right? Remember, sodium does the positive one, excitatory. Excellent. So it's going to be potassium either leaving the cell or chloride entering the cell, right? So those are the two that are going to be involved in that. Now, remember when we were talking about the neuromuscular junction, we talked about how one neural action potential meant one muscle action potential. But that's not typically how the nervous system works. A single neuron in the nervous system can receive somewhere between 1,000 and 10,000 different inputs. A single neuron can have 10,000 different cells communicating with it. So in our nervous system, one excitatory postsynaptic potential is typically not enough to get it to reach threshold. Instead, basically, what a neuron is an accountant. And I don't mean like a stripper on TikTok. I mean a real accountant. Their job is to count beans. All right? You've got 10,000 inputs coming into a neuron. And if 9,000 of those inputs are excitatory and only 1,000 are inhibitory, do you think that neuron's likely to fire an action potential? Let me say that again. A cell has 9,000 excitatory postsynaptic potentials being produced. It's getting 9,000 excitatory inputs and only 1,000 inhibitory inputs. Do you think that cell is likely to fire an action potential? Yeah, absolutely. If conversely, I'm getting 7,000 inhibitory inputs and only 3,000 excitatory inputs, do you think that it's likely to fire an action potential? No. And that's what it is. It counts beans. It's like your checking account, right? You have money coming in and money coming out. And $1 coming in is just as big as $1 coming out. So if you get 5,000 excitatory inputs and 5,000 inhibitory inputs, you're receiving a ton of information. And are you going to do anything as a result of that? You think you're going to fire an action potential? No. Right? 10,000 pieces of information coming in, 5,000 positive, 5,000 negative, and they basically cancel each other out. One inhibitory postsynaptic potential basically cancels the effect of one excitatory postsynaptic potential. So our goal is to count beans. And if our bean count gets big enough where we reach threshold, then we get to fire an action potential. Now, I love the analogy in your textbook. Let's cheat and go to the whiteboard for a second. I love the analogy that your book uses for this. Threshold is basically a bathtub, and our job is to fill the bathtub. So we have buckets, and we need to use this bucket to fill the bathtub with water. The problem is you don't have your stopper. There's no stopper, so you have that little drain at the bottom. So if my bathtub is on the second floor of my house and my hot water faucet is in the basement, I fill my bucket with hot water in the basement. I walk up the stairs to the second story. I dump it into the bathtub. I then have to go all the way down to the basement, fill the bucket, come all the way back up. By the time I come up, what's happened to the first bucket of water?
Cooled.
Yeah, well, not only is it cooled, but if I don't have a plug for the drain, it's gone, right? Hasn't it drained out by the time I've done that? Yeah, so what I need to do is I need to get a lot of buckets close in time to be able to reach threshold. So my goal is to fill this bathtub. Let's say it takes 10 buckets of water to fill this bathtub. I'm not going to be able to run downstairs to my basement, run back up to the second flight, and dump it in there and be able to ever fill that bathtub. However, if I have the faucet right next to the bathtub, and me working by myself, if I just keep filling and dumping and filling and dumping and filling and dumping, and I do that as fast as I can, am I going to be able to fill this bathtub by myself? What do you think? If I can go fast enough, remember, it's a small drain and I have a big bucket. If it's a small bucket, then yes, it's going to drain out too quickly. But if I have a large bucket that I can dump in here and I can do it fast enough, I'm going to be able to fill this up. So when it comes to filling this, when it comes to reaching threshold, I need many signals close in time. Many, let's be more specific, many excitatory postsynaptic potentials close in time. And there are two ways that I can do this. One way, as I mentioned, is me doing it by myself. That would be an example of what we call temporal summation. In temporal summation, it is me dumping those buckets as quickly as I can into that bathtub. Or in this case, temporal summation would be one neuron firing rapidly to produce many excitatory postsynaptic potentials close in time. So if I stand there with my bucket by myself, and I do it fast enough, I can fill that bathtub. But I don't have to fill this bathtub by myself. I have friends, and I can grab 10 of my closest friends, and we can all stand around this bathtub, and we can all take a bucket, and we can all dump one bucket into this bathtub at the same time. So all of us working together in the same location can all dump our buckets into that bathtub at the same time, right? I can't do that now because of coronavirus, but if I can get everybody in the same location, spatial summation, if I can get everyone in the same location, then by getting all my friends together in the same location, everybody with their bucket of water, we can all dump it at once and we can fill the bathtub up and we can reach threshold that way as well. So spatial summation is where multiple neurons all fire an action potential at the same time. Or more specifically, let's do this. All multiple neurons produce an excitatory postsynaptic potential at the same neuron. And in that same location, we can then get it to reach threshold. Now, again, my picture of my bathtub isn't the best, but we got these great pictures from your textbook. So let's take a look at this. Here, we see an example of temporal summation. Again, let's do a really simple illustration of this. Here is our graph. Here is our time. Here is our voltage. As we know, when I release neurotransmitter, I open some voltage gated channels and let's say sodium rushed in, and that would give me a rush of sodium that comes in. We know it's not going to stay in forever, so it's going to eventually go away. And what did we say the average size of a graded potential was?
2.
Yeah, about 2 millivolts. And we know threshold is 10. So that's not going to get it there. That's not going to have any effect. And if we wait 5 minutes and stimulate it again, then nothing happens. And we wait 5 minutes and we do it again, then nothing happens. But if immediately after as we're producing the first, we are able to add a second, and we are able to add a third, and we are able to add a fourth, can we get it to reach threshold that way? By firing multiple action potentials, producing multiple excitatory postsynaptic potentials. Can I add those postsynaptic potentials together to get it to reach threshold? Absolutely. And that is what temporal summation is. One neuron firing fast enough, producing enough excitatory postsynaptic potentials to get the cell to reach threshold. And spatial summation is essentially the same thing. The only difference with spatial summation is instead of one neuron producing all of the graded potentials, one neuron produces one graded potential at the same time that a second neuron produces a graded potential, at the same time that a third neuron produces a graded potential, at the same time that a fourth neuron produces a graded potential. And by having all of these cells all fire and produce graded potentials at the same time, at the same location, we can add those up to reach threshold. And so, like I said, with 10,000 synapses on a neuron, it can be receiving hundreds to thousands of inputs at any one time. And it adds the positives, and it adds the negatives, and whatever the bank account is at the end, if it reaches threshold, we get an action potential. All right. Questions on that? Excellent. All right. Last thing we need to talk about for the lecture, and then finally we'll be able to get to the labs, is our neurotransmitters. There have been well over 100 different neurotransmitters that have been identified in the body. And I'm not going to rattle off all of them for you, but there are some that it is worth spending some time talking about. So let's talk about some of the more common ones. Now, one of the tricks with neurotransmitters is that neurotransmitters are excitatory or inhibitory depending on the postsynaptic cell. As we know, at our neuromuscular junction, when communicating, here, hold on. All right, our synapse communicates with that cylinder-shaped skeletal muscle cell. What neurotransmitter did we say was going to be released at that neuromuscular junction?
Acetylcholine.
Acetylcholine. And remind me again, is it excitatory or inhibitory?
Excitatory?
Excitatory. Right? When it fires, it causes the muscle cell to contract. It's excitatory. It causes it to contract, absolutely. Right? Making the muscle cell contract. Well, guess what? We have other neurons that release acetylcholine as well. And in this case, they release acetylcholine not on skeletal muscle, but on cardiac muscle. There's my branched cardiac muscle cell. And if you were to take a little bit of acetylcholine and sprinkle it on cardiac muscle cell, did anybody know what would happen to the heart? But what happened to the heart if you sprinkled acetylcholine onto it? Anyone know?
Speed up.
You would think it would speed up, but in fact, it actually slows the heart rate down. If you sprinkle acetylcholine on the heart, it is actually inhibitory. How is that possible? How is it possible that the same chemical in one case can cause action potentials by being excitatory and can others slow the heart rate down by being inhibitory? How can one chemical do both of those things?
Different postsynaptic channel.
Exactly. In our skeletal muscle, the channel that is opened on our skeletal muscle by acetylcholine, as we know, is a cation channel. And what's the most common cation that passes through that channel? Yeah, sodium, right? We know potassium flows through it as well, but not as much. However, on our cardiac muscle, it is a different type of channel. And on our cardiac muscle, it actually opens a chloride channel. So when you open a chloride channel, chloride enters into the cell, inhibits it, and it slows the heart rate down. So notice when we talk about the effect of a neurotransmitter, the neurotransmitter itself isn't what really determines the effect. It's really the channel, the type of channel that it opens. And that's how something like acetylcholine can be both excitatory and inhibitory, because it can open different types of channels. All right, so let's talk about some of our, and then let's actually, hold on, I want to go back to that picture. As we've also talked about, we can modify communication. We want to be able to modify communication at these synapses. And there are a couple ways we can do this. One of the ways we can do this is obviously produce more or less action potentials, right? So as we produce more stimuli, we're going to get a bigger signal. Fewer stimuli, we get a smaller signal. That's the obvious one we've already talked about. But we can also release more or less neurotransmitter. We can have more vesicles binding and release, or we can make more neurotransmitter and put more neurotransmitter in the vesicles. But it's not just on the presynaptic cell side. The other way we can modify communication here is by having more or less receptors. If you think about it, if we were to take a cell and we were to put 1,000 receptors on it or 1,000,000 receptors on it, then even a small amount of acetylcholine is likely to find a receptor, bind to it, and cause an effect. However, if we reduce the number of receptors, then we are going to desensitize it. And in fact, if we remove all of the receptors, you could dump a whole bucket of acetylcholine on there, and are you going to have any kind of effect? No. In that case, we would actually say we had desensitized the cell. Right? So we can habituate the cell, make it harder to have a signal, or we can make it more sensitive. So we can desensitize it or sensitize it by also changing the number of receptors that we have as well. Again, the whole key to this process is that it is modifiable. We can produce more action potentials, we can make more neurotransmitter, we can put more neurotransmitter into the vesicles that we release. Some of you had this for one of your essay questions on the last lecture exam. One of the ways we can affect the synaptic cleft, the question was, what happens if we remove acetylcholinesterase from the synaptic cleft? What happens in that situation? Come on, at least I know some of you had that for a question. What happens, even if you didn't? Someone answer the question for me. What happens at the neuromuscular junction if we remove acetylcholinesterase?
There would be more acetylcholine to open up the channels.
Yeah, the acetylcholine stays in the synaptic cleft for a longer period of time. Does it stay in the synaptic cleft forever? No, it's still going to, as Haley pointed out, be able to diffuse out. Some will still be endocytosed, but without the enzyme to break it down, it stays in that synapse, in the synaptic cleft for a longer period of time and has a bigger effect. So it's going to cause a longer, more sustained contraction. Again, is it a rigid paralysis? Does it say locked up forever? No. It will eventually relax, but it'll take a lot longer. So we have a much bigger effect. And then again, also, we can change the number of postsynaptic receptors. All of these are ways that we can change how strong a chemical synapse is. Again, we can't do this with electrical synapses. Electrical synapses are hardwired. They're not modifiable. But this one we can modify. We can strengthen, we can weaken. And again, this allows us to change and modify our behavior. And then, of course, we have also talked about toxins that can block curare, tetanus, things like that, that either can block or can activate the receptor, mimicking or blocking the influence. And we'll talk more about that when we get to the autonomic nervous system. All right, let's quickly mention some of the different types of neurotransmitters. Acetylcholine is the most common neurotransmitter in the peripheral nervous system. So if you randomly grab a neuron in the peripheral nervous system, it most likely releases acetylcholine. And again, remember, as we saw, acetylcholine has the potential to be both excitatory or inhibitory, depending on its receptor. Of course, if it's the most common in the peripheral nervous system, then that means there must be a different most common one in the central nervous system. And in our central nervous system, the most common neurotransmitters are in central nervous system. The most common excitatory neurotransmitter is glutamate, and our most common inhibitory is gamma aminobutyric acid, which in this case is a term I will not make you memorize. You may just simply use the abbreviation GABA. So the most common inhibitory neurotransmitter in the central nervous system, you can actually just use GABA. Others are larger type molecules. Many of them are hormones, like epinephrine and norepinephrine. Is there another name for epinephrine?
Adrenaline?
Adrenaline, right? So noradrenaline and adrenaline, or norepinephrine and epinephrine, both of those terms are interchangeable. What are those involved in? Epinephrine, what is that involved with? When do we use that neurotransmitter?
Increases heart rate?
Right. Why? When? When might your body release adrenaline? Yeah, fight or flight. How about when that bear with an axe walks in the room? Right? Because when a bear with an axe walks in the room, you have two choices. Fight that bear with an axe or outrun somebody in the class. All right, so it helps us to deal with stress. Adrenaline, noradrenaline is our fight or flight stress response. What about dopamine? Where is dopamine? When is dopamine released? What does dopamine do for us? Anyone know? We talked about dopamine with Parkinson's, right? When else might you release?
Increases your blood pressure.
I'm sorry?
It can help increase blood pressure.
It can help to stabilize blood pressure, absolutely. Dopamine is a really important neurotransmitter in our central nervous system. We talked about the effects that it has on Parkinson's, but we're also finding that it is a very important mood modifier. A lot of social anxiety disorders and things along those lines are caused by decreases in dopamine levels. So dopamine can be brought up to help in those types of situations. And again, it can affect stabilization of things like heart rate and blood pressure and things along those lines. We have some neuropeptide type neurotransmitters like endorphins and substance P. Most people have heard of endorphins. Where do you get endorphins from? Come on. Exercise, absolutely. Exercise is one of the things that gives you endorphins. What else does?
Sex.
Yeah, there you go. Love, sex, chocolate, right? Well, drugs, again, remember with drugs, those may bind to endorphin receptors and mimic the effect, which is why those drugs can be, pleasurable, taking those drugs because they mimic it. Yeah, endorphins is typically that euphoric high type of sensation you get, like from things like love, from things like chocolate, from things like exercise, right? Substance P, with a name like Substance P, what do you think it is involved in? What do you think the P stands for? We have happy endorphins on the one side, pain on the other. Our pain pathway is one of our least understood neural pathways. But one of the things we do know about it is many of the neurons in the pain pathway use a special neurotransmitter that, because it's in the pain pathway, is called substance P. And then there's some unique things that can be neurotransmitters. ATP can actually be a neurotransmitter. Certain gases can be a neurotransmitter, like nitric oxide, not nitrous oxide, right? That's what you get at the dentist's office or that rave on Friday night. But nitric oxide can be released by some of our cells to control the diameter of our blood vessels. The smooth muscle of our blood vessels is affected by nitric oxide. So we can dilate and contract our blood vessels by doing stuff like that. That's a great question, Haley. But so here's what I would say. How is that not real? If you are having, extreme temperatures or extreme, stretch or damage or injury or cell death or things like that that are occurring, and you're receiving that information about that harmful information, then how is that not real? That's like saying that just because a photon of light hits my photoreceptor in my eye and sends a signal to my brain, that light isn't real. Right? You're receiving a stimulus. That stimulus is sending information to your brain, and you're responding to it as a result of that. I mean, if pain's not real, then nothing is real, right? Because that's how we perceive everything about the world, not just pain, but literally everything we perceive about the world around us is caused by what we call transduction. Transduction is taking some type of outside stimuli and converting it into the language of our nervous system, converting it into an action potential. So touch, smell, hear, vision, right? All of those things. Well, what do you mean by tangible? Can't hear sound. Can't see sound. True. Everybody has a different level of pain, but everybody has a different level of visual acuity, right? Our ability to perceive light, our ability to perceive sound. Here, hold on. I haven't done this I don't think I've done this in this class yet. I wonder if this will work. Oh, I don't have my good microphone plugged in. I wonder if this. Do I sound different when I plug that in now? Or do I sound the same? Yeah, I sound different. Better? Cool. Sorry
That's
about that.
different.
All right. See if this works. Anybody hear that? Here, hold on. What if I do this? What about that? No one hears anything? All right, it might not be working. Hold on. Let's try one more thing. Oops. I'm not there. Here, I want to go here. I want to go here.
Disney Plus, Hulu, and ESPN Plus now bundled together. The greatest stories and characters, the largest streaming TV library, and exclusive sports. Disney Plus, Hulu, and ESPN Plus. Get all three for only $12.99 a month.
All right, you guys hearing that? What about that? Can you guys hear that? I get a couple yeps. Anybody else? Haha, Lori, you're old like me. All right, quick poll. How many people can hear this? I got at least 3 people who have said yes. Some yes, some no. Some can't. This is, and again, so here's what you're going to do. During the next break, you're going to go to YouTube and you're going to click on mosquito sound. Mosquito sound is a 15,000 hertz. The normal hearing range for humans is from about 20 hertz to 20,000 hertz. However, as we age, or as we listen to our earpods too loudly as we age, we start to lose most of our high frequency hearing first. So what happens as we age is that we start to lose our high frequency hearing. And so what some really clever guys in Sweden found is that they were having this issue with a lot of teenagers loitering out in front of their store. So what they did is they took the sound and they pumped it out of these big, huge speakers outside of their store. Mature, adult, paying customers can't hear this sound, but kids could, and it's annoying. And so the younger ears were able to hear it and they wouldn't want to hang out. Of course, then someone even more clever than that ended up using this to make a ringtone that kids could hear and parents couldn't hear the ringtone back in those days. So yeah, so you'll have to check that mosquito sound and see if you're actually able to hear this for yourself. But so again, the point is that we all have different perceptions, levels of perceptions, taste differently. We have some super sensors. So yeah, I mean, I think I guess I kind of understand the point that they were trying to make with that. Did I turn that off? I forgot if I turned that off or not. I don't want to have that keep going off the whole time you guys are in here. You can still hear it. Okay, there we go. I turned it off now. Now it's off, right?
Sounds the same to me.
Yeah, same to me as well. So again, I've got those I won't say old ears. I just have well-used ears. Let's just say that. But so the point I would say is that, again, from a neurological standpoint, everything that we perceive about the world is converted into action potentials. Vision is probably the best example. If we get a chance at the end, we'll talk about this. Most of what you're seeing in the world around you is your brain's interpretation of the light that's coming out of that, right? And again, how do we know this? Because there are ways that we can modify and manipulate vision so that those systems break down, right? We call them illusions. Visual illusions are basically caused by the breakdown of our brain's ability to be able to interpret our surroundings and convert it into something that we can understand. So yes, theoretically, you could say that that is true for pain, but then you have to say that for everything else as well. All senses are that way, right? That was the whole, for again, those of you who have done any philosophy, that's ultimately what Descartes is a great, famous, ancient philosopher. And that was what his premise is, how do I know that the world around me is real? How can I believe anything that I'm interpreting, like pain? How do I know that that is real? And ultimately, his ultimum that he came up with was, I think, therefore I am, right? He has to start with the basis of the fact that I must be real because I can think I'm real. And then based on the fact that I'm real, I can then start making assumptions about the rest of the world. Because if I'm not real, then yeah, all assumptions are off. So I would say the same thing. So again, he's not entirely wrong, but if you're going to make that form of logic, then you have to say that it's true for everything. Then the same thing's true for vision. There's no such thing as light. There's no such thing as sound. There's no such thing as taste or hearing or anything else that way as well. Excellent. All right. I spent too much time on that, but I like neuroscience. Sorry. All right. Excellent. All right. So your book's got a nice table that talks about the different neurotransmitters. And again, these are the main ones that we need to be aware of and we'll talk about. All right. Thankfully, with that, that is everything we need to do for a lecture standpoint today, which is definitely a good thing because we are way behind on our anatomy. So our goal next is going to be to close this, come here. Start working our way through our brain anatomy list. So hopefully you have your list printed out. Hopefully you've already started looking at it, but if not, shame on you, but grab it now. And we will come back after our first break and we will start working our way through our brain anatomy list. All right, it's 9:15 now, so let's restart at 9:30. And at 9:30, I will, thank you for reminding me at the beginning of class, I will start the recording again at that point. So I'll see you in 15 minutes so we can go over our brain anatomy. Let's go ahead and get started. Let me move this over here. So you should have a handout. The study guide looks something like this that has all of your brain anatomy stuff that we are going to be working on and looking at. And we'll talk about the different parts and regions of them. So again, as you can see from the illustration here, we are focusing on the 5 cephalons, as we've talked about. We have our telencephalon, which is the cerebrum, the diencephalon, mesencephalon, metencephalon, and myencephalon. When we look at the anatomy, the largest part is the telencephalon. The telencephalon forms this structure that is the cerebrum. The cerebrum is the telencephalon. Those two terms essentially are interchangeable. All right, and here we see in a cross section, and again, all of this stuff up here is the telencephalon. This thing that looks like a duck head is the diencephalon. The little mid part here is what is the mesencephalon, and as we mentioned, is also known as the midbrain. Then 2 enlargements of the pons and the cerebellum is the metencephalon. And then this down here is the myencephalon. If we take a coronal section through the cerebrum, what we see, as we talked about in the brain, is that it is divided up into gray matter and white matter. Now, again, these are formed by the parts of primarily the neurons and the neuroglial cells that support them. And the gray matter, remember, as we talked about, is comprised of the cell bodies and the dendrites. Whereas the white matter forms the axons, is formed, I should say, by the axons. And most of those are myelinated, which is why it gets the name white matter, because myelin, again, is hundreds of layers of phospholipid bilayers wrapped around it from those oligodendrocytes. And phospholipids are fat, and it gives it a whitish coloration to it. Now, notice also here, when we talk about our gray matter, the cell bodies and the dendrites in the central nervous system or in the cerebrum are basically found in 2 areas. We have the outer layer, and that outer layer is what is known as the cortex, or because here we're in the cerebrum, it would be the cerebral cortex. Oops. The cerebral cortex. So all of this superficial material is the gray matter. Right? If you remember when we looked at the cerebellum the gray matter was on the outside as well. It was superficial. Where was the gray matter in the spinal cord?
Deep.
Yeah, it was deep there. Right here, the cortex is superficial. So that cortex, that outer layer of cortex, is that superficial layer forming that outer part. However, notice there are also these colored structures located deep inside of it as well. These structures deep are collections of cell bodies, small collections of cell bodies that are deep in the white matter. And these are known as the nuclei. Nuclei being the plural, the singular would be a nucleus. So again, one nucleus, many nuclei. So when we look at the gray matter in our cerebrum, it forms the cerebral cortex and it forms the nuclei. White matter, remember in the central nervous system, forms what we call tracts. Remember outside it forms our nerves, but inside it is tracts. So these tracts are different pathways for carrying information. And again, if we take a look at this pretty picture here, again, most of these pictures are from your textbook, remember, so I've got most of these pictures straight from your textbook. There are different types of fiber tracts. Notice some fiber tracts are very local. So again, if you think of it from a functional standpoint, if I'm trying to manipulate something with my hand and feel what it is, does my pointer finger need to know what my middle finger are feeling? Do I need to coordinate that information to figure out what it is that I'm holding in my hand? Yeah, absolutely, right? At the same time, we can have some longitudinal tracts, right? Because if I'm holding something in my hand and trying to figure out what it is, not only can I use my fingers to feel it, but I can use my eyes to see it and put that information together. And lastly, we have, let me move this down here. We have what we call commissural tracts. Commissural tracts involve fibers that go between the hemispheres. Because, as you guys know, at the end of the lecture, when I'm done, you guys all get on camera, all stand up, and all give me a nice big, huge round of applause, right? And if you think about it, to be able to coordinate that, where you take your two hands and you bring your left hand and your right hand together, you need to obviously be able to coordinate that information. So having axons that go from the left side of the brain to the right side of the brain are going to help. So in our tracts, we have our fiber tracts, we have arcuate, we have longitudinal, and we have commissural tracts. All bundles of axons inside the brain carrying information from one place to another. All right. Questions on that? So we understand what white matter is. We understand what gray matter is. Let's do some other anatomy. Notice, and let's see what do I have next. Let's go here. Let's go back to this picture. This cerebrum has many invaginations in it. It is a big folded structure. Why is it a big folded structure like this?
More surface area?
Exactly, right? One of the advantages, right, you have a piece of paper, it takes up a lot of space. But if I crumple it up into a ball, right, it takes up much less space. And so that's what we've done here. And much like we saw in the cerebellum, we have these nooks and crannies. So if you remember correctly, we have raised ridges and we had invaginations here on our cerebellum. And on our cerebellum, what did we call the raised ridges?
Gyri?
Well, not on the cerebellum we didn't. What did we call them on the cerebellum? Do you remember what we called them on the cerebellum?
Oh, folium?
Folium, excellent. Folium was the raised ridges. And then as Haley pointed out, the invagination, the fold in between, we called a sulcus. Well, notice we have raised ridges and we have grooves on the cerebrum as well. And those grooves are indeed called sulci, or the singular being a sulcus. That's the same. So are the raised ridges the same as well? Is this a folium? What do you think? Is the raised ridge here was a folium, so is the raised ridge here a folium? Nope. Why not?
Because it's a diary.
Well, it's part of it is it's a different area. Part of it is just because anatomists hate you, right? So yes, they've given it a different name. Here on the cerebrum, it is a gyrus. So in our cerebrum, again, we have raised ridges that are going to be a gyrus, and an invagination, which is going to be a sulcus. They are awful, awful people. And some of these invaginations are going to be really, really deep. So deep that we're not going to really call them a sulcus anymore. So let's again, this is a gyrus. This is a sulcus. But a really, really deep sulcus, they have a fancy name for. And what's that fancy name for the deep sulcus?
Fissures.
Fissure, exactly. So we have gyri, sulci, and fissures. These are the morphological structures of our cerebrum. Now, there is a picture here of a cerebrum. And does everybody in the class's cerebrum look exactly like this picture? No, of course not. But what is surprising is actually how similar our cerebrums actually are to each other. We know this thanks to an individual by the name of Corbinian Brodmann. Corbinian Brodmann is a very famous anatomist from the late 1800s, early 1900s, who was an incredibly meticulous anatomist. And one of the things that he was interested in is the morphology, the shape of brains. And so what he did is he started with cat brain, and looked at all the nooks and crannies of a cat brain. And then he took a second cat brain. And what he noticed is that there were some areas that had the same shape. And then he looked at a third and a fourth and a 205th. And after looking at all of these brains, cat brains, what he noticed is, you know what? All of these cat brains have this particular ridge in this area right here. They all seem to have that. And since they all had this, he decided to give it a name. And did he know what it did, or if it corresponded with any function or anything else about it? Again, back in those times, you couldn't see what the brains do when they're working, right? They didn't have functional magnetic resonance imaging or things along those lines. So he wanted to give it a name, and it happened to be the first area that he found that was similar in all of these. So guess what he called it? Called it area 1. And guess what he called the second area that he found? Area 2, and 3, and 7, and 44, and so on and so forth. He went through and found all these different regions that were similar in cats' brains. And then he did monkey brains, and then he did human brains. And so when he found these, and in fact, I have a picture here we'll get to in a minute. There we go. These numbers in circles are actually those Brodmann numbers. So we have these Brodmann numbers. And again, I'm not going to make you memorize the Brodmann numbers. But again, if we were in the classroom and you were holding an arm model in your hand, and I said muscle that has the number 17 on it is the sartorius muscle, would that make it easy for all of you to know exactly what muscle I'm talking about? Yeah, so I may mention these numbers when we're working through this material because, not surprisingly, areas of the brain that are similar in shape actually have similar functions. So while his numbers aren't perfect, there is a tremendous amount of similarity between the structures of the cerebrum and their functions. And Corbinian-Brodmann is the one that helped us to be able to recognize and identify this. So when talking about these nooks and crannies, there's a fair amount of consistency in them. And we can use these structures to identify them. Now, let's look at a real brain. And as we look at a real brain, and again, we see that mitten shape to it. The brain, the cerebrum, because again, remember we're focusing on the cerebrum now, is divided up into 4.5 lobes. And the good news is we've already learned this information. What is the bone that covers the front part of my brain? This bone right here, the one that makes up my 5 head. The frontal bone. And notice the frontal bone covers the frontal lobe. What is the bone in the back of the head? Occipital lobe. What is the one that forms the high sides of my skull? Parietal lobe. And this one over here along the side where the temple of my glasses are? Temporal lobe. There you go. We've used this information once, we get to use it twice. We've learned it already, so the different lobes of the brain, here we go. Here we see the pretty picture. Here we see the different lobes of the brain, named after the different bones that cover and protect them. Now, as I mentioned, there's some similarity in these structures, and one of the most constant structures that we find is what is known as the central sulcus. The central sulcus starts basically at the very top of the skull and comes down inferiorly and a little bit anteriorly like this here. That is our central sulcus. And notice the central sulcus is the dividing structure between the frontal lobe and the parietal lobe. Notice we also have a lateral sulcus. Lateral sulcus is basically what divides the temporal lobe from the frontal lobe and a little bit from the parietal lobe. And notice also we have some structures here that are, and let's do it this way. If there is a sulcus, not surprisingly, there is a raised ridge in front of it. And this raised ridge in front of the central sulcus is called the precentral gyrus. And there is a raised ridge behind the central sulcus. So guess what it's called? The postcentral gyrus. So these are some examples of some of the anatomical features you are going to be responsible for. Now notice, when we look at the cerebrum from the outer surface, there doesn't seem to be the same type of anatomical structures that separate the occipital lobe from the temporal and parietal. Turns out they are, they're just deep. We're not seeing them from this view of the brain. So they're in there, but they're not on your list, so we don't care about them. There is one other big structure you are responsible for, and that is the longitudinal fissure. Notice the longitudinal fissure, again, is a very deep sulcus that divides the brain into its 2 hemispheres. We see it here from the top. And notice also while we're here at the top, we can actually find that central sulcus that is going to come down this way and the central sulcus that is going to come down that way. Now, I've looked at many, many brains. It was very easy for me to find that. Is it going to necessarily be as easy for you to find that from this type of view? And again, your lab exams are timed. So the challenge with this is finding these things. And so that's the advantage of using illustrations like this from your textbook that have these colors on them. It's going to be much easier to find the central sulcus on something like this than on something like this. So I will try to use more of the obvious, distinct structures so that it is easier for you to be able to identify them. As I mentioned, the longitudinal fissure goes deep into the brain, helping to separate the brain into two hemispheres. However, the cerebrum into two hemispheres. We see it nicely from this view, but notice if we go back to this picture, notice here in this picture, this right here is the longitudinal fissure, dividing the cerebrum into the two hemispheres. And notice this over here and this over here is our lateral sulcus. That means this is my parietal lobe, this is my temporal lobe. So notice we can see that in this frontal section as well. All right. So those are the structures you are responsible for. And again, in this case, a structure is a structure. The central sulcus, does the central sulcus have a function?
Probably.
Well, I guess you could kind of say that it is the dividing feature between the frontal lobe and the parietal lobe, but it doesn't do anything. All right? It's a characteristic, it's a structure, it's a feature, right? Like the wall, it separates, I guess, my room from my daughter's room, but it doesn't really have a function. So these are things that are just structures, things that I will point at and say, what is this structure? What is this raised ridge? What is this groove? However, in the frontal lobe, is there going to be part of the cortex that is going to have some specific function? Some job that is going to help your brain to do? Absolutely. And so for those in each of our lobes, there are a ton of areas or regions of the cortex that not only do you need to know the name of, but you also need to know their functions. So if you notice as you work your way through this list, we look first at the frontal lobe. You'll see the frontal lobe, in the frontal lobe, there are three regions. There are three areas of cortex that have specific names and have specific functions. All right, so this is going to be important because, for instance, on the exam, I could, for instance, have this picture with a big arrow pointed here, and I could ask you a bunch of questions. Let's start with the ones we know the answer for. Identify the cephalon that I am pointing at. Which cephalon is this?
Telencephalon?
Telencephalon, exactly. Excellent. Identify the lobe.
Frontal?
Excellent. Identify the structure.
Precentral gyrus?
Precentral gyrus. Excellent. 3 questions we already know the answer to. Now, 2 more pieces of information. On the central sulcus happens to be an area or a region that is known as the primary motor cortex. So this area, the central sulcus, contains the primary motor cortex. And guess what the function of the primary motor cortex does?
Processes movement?
Yeah, And you get partial credit for that on the exam. Absolutely. You have the right idea. This is where we produce the control for movement. But what if we're going to move our body through space, what are we using to move? If I'm doing this, what am I using to do this?
Muscle,
What kind of muscle?
skeletal muscle.
There you go. And let's take it one step forward. Notice as we look at this brain, that is the left side of my brain. So what skeletal muscle is it controlling?
Your right side.
Exactly. So the primary motor cortex provides control for the contralateral skeletal muscle. So notice that same arrow. Now there's 5 questions I could ask. Identify the cephalon. What was the answer to that again?
Telecephalon.
Telencephalon. Identify the lobe.
Frontal.
Frontal. Identify the structure.
Precentral gyrus.
Identify the region or area.
Primary motor cortex.
Excellent. Identify the function of the region or area, region/area.
Movement of skeletal muscle. Contralateral.
There you go. Movement of contralateral muscle. Excellent. And there you go. Just that easily. That's what you're going to have to do on the exam. All right. And it's cool the way this is set up. It's not just random. I'm going to move that there, and I'm going to erase my arrow. There is an actual map of the contralateral part of your body here in this on this primary motor cortex. So for instance, down here is where your head is located. And then we have the representation of the hand here with a huge representation of the thumb and the hand, big representation of the face, and then a tiny part of the body, and then the legs, and then the feet, right? There's this whole map of our body here. And while your trunk may be the largest part of your body, it doesn't have the most cortex, right? Because the amount of cortex you have is about how much control you need. You need a lot more control of your face so you can make those subtle facial expressions. Huge control of our hands so we can manipulate the world around us. We don't do much with the trunk of our body. So we don't, we have to stabilize and maintain our balance, but we don't do a whole heck of a lot of it. So it's a relatively small map. So we have this beautiful map of our body here on this precentral gyrus on that primary motor cortex, allowing us to control, right? Well, again, the areas don't look like the part of the body. It's not like the part that moves your head looks like your head, or the part that moves your arm looks like your arm. There's just this map of the body over this region. So again, the parts don't look like it, but I'm telling you that functionally there is a map there. And again, you may not have thought of it in those terms, but let's say there happens to be a very large blood vessel that travels right over the top of our central sulcus. And if one of the blood vessels coming off here gets blocked, suddenly this part of the body doesn't get blood for a short period of time. What do we call that condition? A stroke. And if that part of the nervous tissue doesn't get oxygen and dies or gets damaged, what happens to poor grandpa?
Deficits.
Yeah, now grandpa can't move his right hand or his right arm anymore because the part of the cortex that controlled that hand, that controlled that arm, has been damaged. He can still move his face. He can still move his legs, his feet, his trunk. But in this case, the arm and the hand have been damaged. Or maybe it was down here more, where he has some facial paralysis and maybe paralysis to the hand or something like that, right? Depending on what part of it is damaged is going to determine what the deficit is. And because it only happened on the left, it's only affecting the right side of his body. The right side of his body is fine. There's a picture of this in the book. They call it a homunculus. That's the term they typically use for this picture, this image of the brain. And I think I have a picture that I'll show you a little bit later too, but we have that there as well. All right, so that is our first region, our primary motor cortex. Let's switch to, oh here, I love this picture. We see some nice nooks and crannies. Here the protective coverings, outer coverings have been removed away. Here we go. So here we have those Brodmann numbers that will help us as well. And again, we just finished talking about our primary motor cortex, which is Brodmann's area 4. But again, you don't have to memorize 4, it just helps us to find it. Let's talk about the first region of the brain on our list, the prefrontal cortex. The prefrontal cortex is basically all of this material in the front. Oh, I have a question. Hold on one second. We'll come back to this. Let's see how clever I am. I am not clever. All right, let's make me clever. Give me a second here. Actually, let's just do it this way. Perfect. All right, that works. All right. So we are talking about our first region of the brain, and the first region on the brain on our list is the prefrontal cortex. Again, basically the gray matter here in the very front of the brain. This prefrontal cortex is the part of the brain that is responsible for our intellect, for our reasoning, for our personality. How do we know these things?
When the region gets damaged, they can tell what was affected.
Exactly. One of the best things that ever happened in the history of the field of neuroscience was the development of helmets. Because before helmets, young men went off to war and they died. After helmets, young men came back from war injured. And when they were injured and part of their brain was blown off, we could see what deficits they had as a result of that injury, right? And it wasn't always war, right? One of the probably most famous examples is an individual by the name of Phineas Gage. Who here has heard of Phineas Gage before? Allison, what do you know about him? Just heard the name? There you go, exactly. Phineas Gage was a railroad worker. Specifically, he was a railroad foreman. Foremen were typically very even-mannered, very well-tempered, responsible individuals. And one of the reasons for that is they were primarily the ones that were responsible for dealing with the dynamite, right? When building the railroads, the job was to get that railroad from one location to the other as quickly, as easily, as efficiently, and as cost effectively as possible. So when people came across a mountain, was the plan to build the railroad track all the way around the mountain? No,
You blow a hole in the mountain and you get to go through the mountain, and it's much, much quicker that way. The way that they would remove rock is basically they would drill a big hole down, and then the foreman would take the dynamite and use a tamping rod to tamp that dynamite down to the level that it needed to blow up and have the most efficient explosion possible. And these damping rods were 10-foot metal rods. Well, good old Phineas was tamping it down one day, and the dynamite prematurely exploded. And that rod came up, hit him in the forehead, came out the top of his head, knocking him and the rod back 20 feet. And shockingly, he survived. But the front part of his brain, his prefrontal cortex, was destroyed. And while he survived, it changed his personality dramatically. Like I said before, he was a very mild-mannered, even-tempered individual. And if you read some of the reports, he became the kind of man who would drink during the day and who would curse at women. It changed his personality entirely. And so stories like Phineas Gage, stories like other people who have had head injuries like this, got us to realize that your personality, your behavior was found in the prefrontal cortex. And from a neuroscience standpoint, this was a remarkable finding, because if we could then modify this region of the brain, we could therefore modify someone's behavior. And they did research on animals and things along those lines. And they decided, hey, if we can manipulate this region, we can change someone's behavior. This was a revolutionary finding at the time, right? If you think about before that, if you had some kind of mental disorder, right? You were a female who was a little too uppity. You were a male who liked other males, right? If you were one of these type of deviant personalities, they locked you up in a sanitarium. And was a sanitarium the place they would send you to get better? No, basically they locked you up so you couldn't hurt yourself or anybody else until you died. But now they realized if we could modify this person's prefrontal cortex, we can change their behavior. And that's exactly what they did. What they would do is they would take a metal rod, a hollow metal rod, and that hollow metal rod would either be inserted through the orbit of the eye or into the nasal cavity. And that metal rod would be inserted up and broken through the olfactory epithelium, through the cribriform plate into the brain. And it was a hollow tube. And in this hollow tube, it had a little slit in it. They could then push in a bit of razor wire. And the razor wire would come out, and then they would rotate that razor wire around until your behavior changed. And I do mean until your behavior changed, because the people were alive were awake during this process. They would come in, not surprisingly, nervous about the idea of this happening to them, and they would continue to do it until they were no longer nervous anymore. What did they call this process? Come on, I know you know. Someone just mentioned nurture ratchet. There you go, a frontal lobotomy. This was a frontal lobotomy. One of the most famous doctors who was responsible for this was a doctor by the name of Watson. These hollow tubes were called canalies, and he would have two of them in holsters on his side. And he would do both sides of the nose at the same time. So he would do it with these. In fact, he would tour the country to different sanitariums where he would then compete with the local doctors for speed to see who could do it the fastest. Hopefully you are truly appreciating the barbarianism of this, right? And not only is this incredibly barbaric, but again, you got to remember why they were doing this. At the time, it was revolutionary. The thought that we could actually change someone's behavior and potentially get them back into society, right, was a remarkable finding at the time. They didn't necessarily go about it in the best way. The intentions were relatively good. We wanted to change people's behavior. Again, like I said, though, you got to be careful what they thought aberrant behavior was at that point in time. Like I said, if you were a female who had the audacity to have an opinion on things, or if you were a male or female that liked someone of the other sex, right? Those were examples of some of the deviant behaviors that people would get these lobotomies for. But again, at the time, the idea of being able to modify someone's behavior was truly, truly revolutionary. Problem is, it didn't always work. Yes, some people it changed their behavior, but others it didn't. Others' behavior got worse. And that doesn't even talk about all of the infection and other issues and drainage of cerebrospinal fluid and all the problems that go along with that. And is that why we've stopped doing it? Because we as a society realized how horrible and barbaric this was, and so we should stop doing it because we don't like to do bad things to people? No, why have we stopped doing this?
They found medications to do it.
Yeah, why do I have to bother with all of this pesky surgery when I can just give someone a pill? Oh, Allison, it's got nothing to do with it not working. Just look at the pills. All right, you have some aberrant behavior, I'll just give you a pill. If you're shockingly an 8-year-old boy who can't sit still for 4 hours at a time, we'll just give you a pill to get you to sit down. And if pill A doesn't work, we'll just give you pill B, or maybe 2 of pill C's. Does every pill affect every kid the same way? Every person the same way? No. So we'll just use a different combination of medicines. We don't know what the long-term implication of these medicines are. Many of these medications, we don't know the actual mechanism by which they work. All we know is that sometimes when I give this pill to someone, they behave the way I want them to behave. What we are doing with medication is just as barbaric as what we were doing with those frontal lobotomies. It's just more sterile. I hope in 30 years, we'll be laughing at the medication of individuals the same way we laugh at these lobotomies. But it's just as bad. We don't know how it works. It doesn't affect everybody the same way. We just keep bundling different medications and different combinations till we get the behavior we want without understanding how it's working and what the long-term implications of them are. But as long as we can get someone to behave the way we want them to behave as a society, then that's all that matters. All right, off my soapbox. Questions on that? All right, excellent. Let's go back to this picture and do the third area, the third region we are responsible for in the frontal lobe. That third region, if you look closely, I keep losing my annotation. There it is. Let's use that. If you notice in this area right here in the frontal lobe, there are these 3 parallel gyri. And these 3 parallel gyri are an area that is known as Broca's area. Why is it called Broca's area? Yeah, good old Bob Baroka was the first guy that had found it and described it. And that is where it got its name. However, it is also referred to as the speech area. And guess what the speech area helps us to do? Production of speech, right? It allows us to produce speech. Again, one of the ways that we can understand how an area works is by looking at its deficits. If someone had damage to their speech area and you ask them to raise their left hand, they would still be able to do that. Now I know that's my right hand I'm raising, but you get the point. They can understand speech. They can comprehend speech, but they have difficulty producing it. Some can't produce speech at all, but more commonly, you get a disruption of the normal speech pattern. So instead of saying, Bill and I are going to go to the store to get milk, someone with a Broca's aphasia, and that is the term we use for this, that deficit, a Broca's aphasia instead would say, Bill, the door, milk. Now, some of these deficiencies in Broca's area is not as bad. It doesn't stop you from producing speech, but you may have difficulty with certain words. And what do we call that condition?
Stutter.
A stutter. Excellent, right? Our future president had a stutter issue that he dealt with when he was younger. And one of the ways that people have stutters, how do they kind of teach them to how to work their way around it? Anyone know? All right, let's think of it this way. How many people here have heard Adele sing? Anyone here heard Adele sing before? On iTunes, there you go. Beautiful, stunning voice. How many people have heard Adele talk? Allison, did you understand anything she said?
My kids love carpool karaoke, and we had to watch it like 3 times before we could understand everything.
Yeah. Why?
She has an accent.
A huge, very deep accent. So why doesn't she have that accent when she sings? Well, it turns out the speech area, Broca's area, really is just for speech. Singing is a completely different type of process than speech. So one of the things they actually teach people who have a stutter to try to help them to get over that is to speak in more of a sing-songy kind of voice, because if you can sing, if you can talk in a sing-songy kind of voice, it can get you around that stutter. This Broca's area truly is just specific for speech. That's why you see many singers who may have a very, very strong, thick accent when they speak, but it's not there when they sing, because singing and speech is not the same type of function. So again, these types of processes really show how the brain works. All right? Questions on that? All right, excellent. Just that easily, we have identified the 3 regions we're responsible for in the frontal lobe. Let's talk about the 3 regions we are responsible for in the parietal lobe. The first is on the post-central gyrus. This over here is our primary somatosensory cortex. And guess what our primary somatosensory cortex gives us?
Somatic sensation.
Yeah, our sense of touch. And again, specifically contralateral touch. So contralateral touch on the surface of our skin. Again, we have this representation of the body with the big, huge representation of the head, with a big, huge representation of the hand, smaller representation of the trunk and the legs and the feet and all of that. That same map that is on the primary motor cortex is essentially on our primary somatosensory cortex, which again, as we mentioned with poor grandpa, when he has that stroke, right? Not only does he get paralysis of his hand, but he can't feel everything with his hand. However, is the deficiency always mirror symmetrical? No. He may have no feeling for his entire arm, but may just not be able to move his hand. During the break, I'll find some of these homunculi for the primary motor and the primary somatosensory cortex so we can see what those look like. I have some pictures of them, although I don't think I have them here. So I'll find a good picture of that and I'll share that to you after the break. So again, primary somatosensory is where we get our contralateral touch. All right. The second area is this dotted line area right here. Again, remember, Brodmann did a pretty good job of finding these regions or seeing that these regions corresponded with anatomy, but that is not the case here. I think I have another cool picture of this. This is the one I want. Perfect, excellent. So notice this was our Broca's area or speech area. This area here is what is known as our general interpretive area. And this general interpretive area is also what is known as Wernicke's area. Because good old Bob Wernicke. Now, this general interpretive area, right? Speech center is where we produce speech. General interpretive area is where we process speech information. Again, one of the ways we can see these deficiencies, see the function of these areas is by seeing how they're deficient. Remember, speech area just affects my ability to produce speech. Interpretive area affects my ability to be able to produce meaningful speech and interpret speech as well. And again, one of the best ways to see this is to see this with a deficiency. Let's take a look at this. Hold on.
What's this?
The big anchor right there, end of the football.
End of the football?
It influenced the football right here. The cause that it comes in, see?
Yeah.
The end of it, see? Yeah. Oh, we got all of it, see?
I don't understand.
Can you guys hear this? Okay.
Well, it's pliable in the other, see? You don't understand it, but it's products in the long tree coming from there. And it comes source from the country out through, see? All the under it, see, it comes up, finally come out here and goes out, see? Get it?
Sort of.
Yeah, it comes out here, see? It comes out here, here, and here.
Uh-huh.
And it comes in,
Uh-huh.
and it comes out, and bing. That's a good flow, see?
That's a good what?
That's a good flow goes and fly, see, it flows cold or it gives or kicks out or bing or whatever it is. We has no football about it out. It's just plain, see, but they feel that. I guess you meant you have beds, see, I don't know.
Where do you live?
Where do you live? And you live? You're up here, see,
Where do you live, Doc?
where do you live?
Where do you live?
Where do you live is, well, if it's around here, around here. You?
You used to be a dentist.
Yes,
Where?
right here. rest of the here, yes.
And how many years were you a dentist?
How many? Holy Christmas. Oh, about 100 and, let's see, 130, about 40. No, gosh, what that is it? One plus 5. Wait a minute, no. It's a pretty hard class to get to this one day. It's about every year off, see? It's class, it goes for about over 30, see, or 130 for down, see, in front of out for it's less against her, and then it comes back to the against her 4, and then goes out for the rest of the time. The third is not the serve, like the soldiers, where they have the soldiers and the singers and the suburbs and all the first third level and watch out in the short side and they cross with the sugar. 2 hours, 2 high or low. Wow, God, these are terrible. Wow, these are terrible words.
These are terrible words.
They sure are.
Hard?
Yes.
What you're saying, you mean?
Yeah, that's what they do. They actually don't allow tutor. It's when they hire in hard go. And then when they go out, finally they quit, see?
Notice if you were sitting at a Starbucks and these two people were at the table next to you, and you really weren't paying attention, it sounds like they're having a normal conversation. It has the cadence, it has the inflection, it has the pace. It's only when you really start to stop and think and listen to the words that you can say that a lot of the stuff that he's saying doesn't make sense. And part of the challenge here is that he's having difficulty saying things. You see this at the end where he talks about how hard these words are, right? He's all like, it's okay in here, but then they come out and they die. But part of it also is problems with his ability to interpret information. Notice she asks him, where do you live? So if I look you in the eye and I say, where do you live? You, as a person who has to think about that, do I really want him to know where I live? Would repeat the question, but you turn it around. I say, where do you live? And you say, where do I live? Right? Because you're trying to think, do I really want him to know? Right? You turn the question around. She says to him, where do you live? And he repeats it identically. Where do you live? He's repeating the words because he's trying to make sense to it. He can't interpret. He can't make sense of the words. Obviously, some of the words he understood. She asked him if he was a dentist and where, and he pointed to his mouth and laughed about that. She also asked him how long he was a dentist, right? Just looking for a number. And he spent practically 5 minutes sitting here trying to work that out. And I've watched this many times, and it looks like what he is trying to explain is that he became a dentist, went away to war, came back from war, and has been a dentist all the way through there. So he's talking about the soldiers and around the backside and the marching and all of those kind of things. So he's trying, he understands some of the questions. He's trying to answer some of the questions. Some of it doesn't interpret. And that's one of the challenges with these deficiencies. It's hard to ask this person, how much of what I'm saying do you understand and how much of what I'm saying do you not understand? Because there's that difficulty in communication both ways. Like with many types of cognitive disorders, it's often much harder on the family than it is on the person itself. He's clearly aware of the fact that he has a deficiency. Like you talked about, it's hard to get those words out. They come out and they just die. But how aware he is of that, it's not fully understood. We're not fully able to tell that. Now, there's one other part of these two areas that I want to talk about that is really, really interesting. We've talked about speech area and so Broca's and Wernicke's areas, the speech area and the general interpretive area. But as we've also kind of hinted at with our somatosensory and our primary motor cortex, is there is lateralization. What happens on one side of the brain is different from what happens on the other side of the brain. And we see that with the speech area, and we see that with Wernicke's area. For right-handed people, over 90% of right-handed people have the speech area just on the left side of their brain. And even for left-handed people, it's around 70%, about 75. For left-handed people, have it on the left side of the head. For left-handed people, about 15% have it on the right, and about 10% have it on both sides. Now, the question is, how do they know this? Again, we could easily study this now because we could put you in one of those big magnets, do a functional magnetic resonance imaging. And what a magnetic resonance imaging does is it looks at brain flow to different parts of your brain. So you ask you to speak and they see an increase in blood flow going to the left side of your brain in that Broca's area, and we know it's on the left side. If we saw an increase in blood flow to the right side, we know it's on the right side. And if we saw equal blood flow to both, we know it would be both. But we've known this for hundreds of years. And the way they used to test this is they would actually inject an anesthetic into one of your carotid arteries. And that would basically put one side of your brain to sleep. And so what they would do is they would inject you on the left side and ask you to say your name. And if you couldn't say your name, it's because they put the left side of your brain to sleep, and that's where your speech area was. The same thing is true for your general interpretive. Most people's general interpretive area is, again, on the left side. So then the question becomes, what's on the right? And it turns out for the general interpretive area, it is a spatial awareness. Again, one of the ways we can tell this is by deficiencies. I actually had a student, gosh, a couple of years ago in my class whose boyfriend had been in a car accident and damaged the right side area where this spatial awareness is. And if this occurs, what happens is people lose perception of the contralateral side of the world. So what would happen is you would show this person an image of a flower in a flower pot, and this is what they would draw. They would just draw the right side of the flower. They don't pay any attention to the left. When they shower, they don't wash the left side of their body. When they get dressed, they don't put clothes on the left side of the body. During the day, they will get angry at that person who is so close in their personal space on the left side of their body. They can lose awareness of everything on the left side of the world. So they lose that spatial awareness as a result of that. So we have this lateralization of our tissue. Not only is it just that specific region of cortex, but it's just this select region of cortex on the left side of our brains. Again, our nervous tissue is highly unique, highly specialized. Location, structure, function. All right. Questions on that? Excellent. From here, let's go back to this picture and let's talk about the occipital lobe. The occipital lobe is probably the easiest one because, and again, I'm biased, I'm a visual neuroanatomist, but we are visual organisms. Over 40% of the information that we get from the world around us comes through our vision. Each optic nerve has over 1 million axons in it. And over 60% of our brain is involved in processing visual information. And a lot of it starts right here. This area right here on the very back, very back of the brain is area 17. And that is our primary visual cortex. That is where we perceive information on vision, on light. Now, this is just where we get light. And again, if you damaged your primary visual cortex, what would happen? If you damage your primary visual cortex, what do you think would happen? Nope, sorry, I don't have my chat window open. Yeah, and not just visual issues, you'd be blind. You would not perceive any light at all. Nothing but darkness if it was destroyed. Now, hopefully you haven't had your primary visual cortex destroyed, but maybe you did take a blow to your head. And as you took a blow to your head, your brain pushed against the back of the skull in the back. And what happens when that occurs? I'm glad to hear that many of you haven't been punched before, but yeah, you see stars, absolutely. You see flashes of light. Those flashes of light are from the nervous cortex pushing against, depressing against the skull. And so, yeah, you see those stars, you see flashes of light as a result of that. But that's all it is, it's light. Our primary visual cortex is where we get the basic information. But then once we get that basic information, we have to process it and make sense of it. Oops. And to do that, we need our association cortex. Now, for vision, that's a large part of our brain. And the different areas have different levels of organization. Let's start simple. Area 18 right here is the area where we make sense of color. So notice if you damaged 18, you would still be able to see lights. You just wouldn't be able to see color. All right, it's where we process and make sense of the information. In fact, we have two big pathways. One big association cortex pathway that kind of goes up onto this part of the brain, and one big association cortex that goes off onto this part of the brain. The top one up here is what we call the where pathway. This is where we process different types of information about where things are in space. Doesn't matter what it is. It could be a dot. It could be your grandmother, whatever it is, where it is, and how it moves through the world. Again, one of the ways we can really appreciate how this works is by understanding deficiencies. There is an area up here, and you don't actually have to know the name of this area. I'm just going to write the initials for it here. It's abbreviated MST. And again, you don't need to know this. I'm just using this as an example. Up here is a region called MST, and MST is what provides us with our optic flow. Optic flow is how you see the world in space, right? As you move through the space, oh, hey, I can demonstrate this, right? Going towards something, going away from something, rotating. As you see that, the world moves. So as you walk through a classroom or walk through a room, you see the world flowing around you or rotating as you turn or moving away from you as you go back. That is our optic flow. There are people who have injuries to their MST area and they can't see or perceive movement. Now that may be hard to understand, but as we mentioned, when you're at that rave on Friday night having your nitrous oxide, one of the things they may do is put on a strobe light. What's it like when you're when someone has a strobe light on?
Kind of like still pictures of movement?
Yeah. Instead of seeing me wave my hand back and forth, you'd see my hand here, and then suddenly it's there, and then suddenly it's there, and then suddenly it's there, and suddenly it's there. That's what it's like to have a deficiency to MST. It's like constantly living in a strobe light. You see no movement. You just see a bunch of static images. Can you drive a car if you have an injury to your MST? No, absolutely not. Can you pour yourself a cup of coffee? Coffee's here, coffee's suddenly overflowing. No, but guess what? They do pour themselves a cup of coffee. How do they do it?
Over the sink?
That's not a bad guess, but then you waste coffee. Slowly could work as well, or you put your thumb in the mug. Put your thumb in your mug, you can pour until it comes up to your thumb, and then you can do it blindfolded, right? So yeah, people are clever. People are pretty ingenious on how they deal with these, but it is, again, if you can imagine what it'd be like living in a strobe light, that's got to be an exhausting deficit. The bottom part of this pathway is what we call the what pathway. What pathway is about identifying what an image is. There is an area down here in temporal, or what is abbreviated IT. And this IT area allows us to identify objects. There is a very, very classic case of a very famous, I'll explain this in a second, Laura. A very famous example of an individual who has an IT deficit. And in one of the classic examples, they showed him a glove. So he was able to recognize the glove. He recognized that it had the 5 extensions that were coming off of it. It happened to be blue and yellow striped, and he could identify the stripes, and he could identify and describe the characteristics to it. He could see all of the characteristics of them. But then when they asked him what it was, he didn't know what it was. So he sat there and thought and thought and thought and thought. And then looking at the extensions on it, he thought, that must be a coin purse, right? One of those extensions are for pennies, and one's for nickels, and one's for dimes, and one's for quarters, and so on and so forth like that. They then hand him the object. And once he can feel and add that extra sense to it, then he's able to recognize it as a glove. But just by viewing it, he can't recognize it. And it's not just objects. He's married. But if his wife is standing still, he does not recognize her. The second she speaks, he can recognize her. Sometimes when she's walking, her movement can help him to recognize her. But if she's just standing still, he cannot recognize her. There have been several books written on him. One of the most famous ones is The Man Who Mistook His Wife for a Hat. All right, this was this individual who had this IT injury. Do you need to know IT and MST? No. So to answer your question, Lauren, no. But the point I'm trying to emphasize, because again, it's these deficiencies that help us to understand. What association cortex does is to help us to make sense of the basic information. Primary visual cortex is where we perceive light. And these big two huge pathways, the what and where pathway, is where we make sense of that information. And that is what association cortex does. Association cortex makes sense of this information. So while you don't need to know any names of them, knowing that there is a where pathway and a what pathway that are part of the association cortex is something that you'll need to know. And guess what? It's not just vision that's like this. Hearing is like this as well. Notice on the temporal lobe, we have an area that is the primary auditory cortex. This is where we perceive sound. And then we have the association cortex around it, where we make sense of that sound. So again, primary auditory cortex is where we receive that sound information. Association is where we realize it is an alternating frequency that is increasing in amplitude, and we process that information and go, oh, that is a siren and it is coming towards me. I need to get out of the middle of the road. So the primary cortexes are where we receive the information. The association cortex is where we make sense of that information. All right. Questions on those? All righty. Let's go ahead and take our next break. And when we come back from this break, we will talk about our fifth lobe. Because again, I not so subtly before when we were talking about a lobe said 4.5. There is kind of a half lobe, or what some people think of as a fifth lobe. And that is what we will talk about next. And that'll be a good lead into the next part we need to talk about. But let's come at this fresh. Now is a good time to take our next break. We'll take a 15-minute break. That means coming back at 10, looks like 57. Oops. And we will restart at that point. And I'll start the recording. All right, any questions before we take our next break? All right, during this break, I'll see if I can find that homunculus picture that I was talking to you guys about. All right, see you in 15 minutes. All righty. I found a couple quick pictures that I just did a quick web search and found and wanted to show you. This is a really nice one that shows the two areas and that, again, that representation of the cortex for the different regions for both motor and for sensory, for motor and both sensory that we can see here. And again, remember, the size of a region is not necessarily indicative of how much of the cortex is involved in processing that information. The more cortex we have for processing the information, the more sensory receptors we have for it, and the better our discrimination is. And like I said, this representation is also often referred to as a homunculus. I found this, again, it's not the best resolution image, but you see many, many images of this, of people who have done artistic representations of what the human body would look like if our size of our body was based on the cortical representation that we have. So you can see that representation there, again, both for the sensory and for the motor. So we see those as well. All right. Here we go. Now, the last part of the telencephalon that we need to talk about for today is the area collectively that is known as the limbic system. I have written down my definition of this here because I wanted to be able to speak it specifically in the way that I describe it. So the limbic system is basically between the cerebrum, the cerebral cortex, I should say, and the diencephalon. So it is in the white matter mostly. But here is the thing that I wanted. It is a functional group that is responsible for emotions, memory storage and retrieval. And here's the part that I wrote down because I wanted to make sure I got it. And linking conscious thought and unconscious functions. So let me say that again. It links conscious thought and unconscious functions. What do I mean by that? What do I mean that it, I'm sorry, go ahead.
Habits, like people unconsciously bite their nails or play with their hair and stuff?
That's actually a really good example. Absolutely. That would be an extreme example of it, but you absolutely have the right idea. Does what you're thinking about, do your emotions and or what you're thinking about affect your body physiologically? Yes, it could be the way you look or the manner that you have or the biting of your nails, but it also could be an increase or decrease in your heart rate. Right? This is one of the things that poker players look at. You know, one of the things that poker players do is they wear, when they're playing, they wear the dark glasses, they wear their hoods over their faces so that they're not giving anything away. Because one of the things that a poker player will do is he'll look at your carotid artery, he'll look at your neck. Because if you are bluffing, making a big bluff, or maybe you get your cards and you get really excited about the hand that you have, then that excitement increases your heart rate, increases your blood pressure, and you can actually see the carotid artery kind of palpating as a pulsating as a result of that. So these types of things, and again, this is the thing that the FBI with their lie detector tests are looking for. How much sweat are you producing? What is your heart rate? What is your blood pressure? How much are you wiggling around in your chair? Our conscious thought, what we're thinking about, what we're feeling, affects our body physiologically. And this limbic system is responsible for that function. Now, it starts deep here in our lateral sulcus. Notice if we were to take a pry bar and lift up our lateral sulcus, deep inside the lateral sulcus is a bit of gray matter, a bit of cortex. Another good way to see this is to take that coronal section. Remember, we looked at that coronal section before, and here in this coronal section, here we saw the lateral sulcus, and notice there's this big fold of gray matter inside the lateral sulcus. This big fold of gray matter is what is known as the insula. There are some anatomists who think of the insula as a fifth lobe. So that's why I said 4.5 before. Again, you can call it a lobe, you don't call it a lobe, I don't care either way. But this insula is the cortex associated with all these nuclei in here. And the insula and these nuclei together make up our limbic system. In particular, our insula plays an important role. Our insula over here plays an important role in our desire, our pain, our anger. But one of the things that they're looking at the insula for a lot right now is addiction. One of the big questions is you can have 15 people smoke a cigarette. Just one cigarette, right? 5 of them may walk away thinking it was a horrible experience and never do it again. 3 of them, right, are going to like it and maybe be willing to try it again. But 1 or 2 of them is going to be instantly hooked, love it, and basically become a 2-pack-a-day smoker for the rest of their lives. And it's not just things like nicotine or drugs, right? Sex can be an addiction. Gambling can be an addiction. Gambling is one of the easiest ways to study this, and this is what they've actually done. What they will do is they will put you in one of those functional magnetic resonance imaging chambers that we talked about to look at blood flow, and they'll have you guess the results on a coin flip. There's going to flip a coin, and you're going to guess whether it's heads or tails. And you're going to see differences in blood flows in individuals who do that. But what's interesting is someone who is addicted to gambling will see a tremendous amount of activation in their insula, whereas someone who is not addicted to gambling will not. Again, as we've talked about, this can be powerful information. If we were able to find a way to block the activity of the insula, it might be possible to curtail addictions that way, whether it's sex, whether it's drugs, whether it's gambling, whether it's smoking. If we could block the stimulus of the insula, that may help to alleviate that strong desire to continue that behavior. And it may be a way, a very simple way to block addiction. So it's a really, really interesting field of study that they're looking at right now. There are some other important structures in here as well. Notice this is that coronal view. Here we have a partial sagittal view. It is a sagittal cut through the top part, the parietal and frontal lobe, but notice the temporal lobe is intact. The reason the temporal lobe is intact in here is because here in the temporal lobe are some of these important structures. So again, notice here we see it with this one right here. This almond-shaped structure is a structure known as the amygdala. The amygdala, or also known as the amygdaloid body, both of those are acceptable terms, is what plays an important role in memory, and specifically in our interpretation of emotional stress, especially fear and anger. We as humans are very, very good at interpreting people's facial expressions. Right? You come stumbling home drunk at 3 o'clock in the morning, fumbling with the keys, trying to get into your house, and your significant other has to come and open the door for you. And without them saying a word, just by looking in their face, can you see how proud they are of you and how aroused they are by your presence there? Absolutely, right? You can tell exactly what they're feeling just by looking them in the face, right? We are very good at interpreting facial expressions. And monkeys are really good at this as well. Monkeys have a very, very visual communication, and they communicate a lot with facial expressions. And they have two main types of facial expressions. They have an aggressive facial expression, and they have a submissive facial expression. And monkeys are really good at interpreting these. They will sit a monkey in a chair, and they'll give the monkey 2 buttons, and they will show him a bunch of pictures. And if it's an angry face, they push the angry button. And if it's a submissive face, they push the submissive button. And when they do this activity, they get a little sip of juice as a reward. And the monkeys love juice, so they will sit there for hours and do this activity. And they're really, really good at it. Unless they have damage to their amygdala. If they have damage to their amygdala, they have difficulty interpreting the differences between the fear and between the submissive. The amygdala is also, and this is what we use, the term we use in our house, the amygdala is also your worry monster. All right? The amygdala's job is to keep you alive, make you aware of things that are scary. But the amygdala lies. The amygdala is the thing that tells you when you hear that creaking of the floorboard that it is the monster coming out from under the bed to get you. Or that car door that slams outside, or the strangers that are going to break into your house and steal your mommy and daddy. All right, this is your emotional fear center. It's important evolutionarily because it helps to keep us alive, but it can tend to exaggerate those fears. Right. So one of those things you have to worry about, and get, especially with little kids, is getting out of that limbic system, getting out of that worry monster and getting into the frontal lobe. So for those of you who have had anxious kids, and my youngest was a little bit anxious when she was younger, she still is a little bit, there are all these tools they give you, like tricks to get you out of the amygdala and get you into your thinking brain, right? Like to come up with an animal for every letter of the alphabet. Or to look around the room and think of something for every color, find something for every color in the rainbow in the room. Or count back from 300 by threes, right? All of these types of activities can help them to get out of that worry monster, out of that limbic brain and into that frontal brain to be able to find that. No, great question. The limbic system, so the limbic system is all of these structures in here, the individual nuclei, and the insula is the cortex that basically controls the limbic system. So this right here is just the insula, but all of these things together are the limbic system. And one of the structures of the limbic system is that amygdala. And here we see the amygdala from this view as well. This right here is that amygdala or amygdaloid body. Another structure that is found here in the limbic system, and again, we see it in this nice view, although they didn't do a very good job of really showing it to us. I'll explain in a second. This big globby red thing right here is a structure known as the hippocampus. It is called the hippocampus because it's not a big globby thing like that. What actually happens is it has a body to it, and then it comes down to a tail that curls around. All right, so this is what it actually looks like in that glob. And that's where it got its name. Hippocampus means seahorse. Think about it, if you put a head on this, this would look like a seahorse with the body and the curved tail. The job of the hippocampus is to convert our short-term memory into long-term memory. All right? I give you 3 items: ball, boat, cat. Repeat them back to me.
Ball, boat, cat.
There you go, exactly. Right? And if I asked you to do it again now, could you do that? Ball, boat, cat. Excellent. Right? And pretty much anybody can do that because you're keeping those items in your working memory. However, someone who has damage to their hippocampus, you ask them to remember those 3 things and they can say it right back to you as well. But then you give them a sheet of mathematical questions, addition and subtraction problems that they have to fill out as quickly as possible. Then as they get done with that, you ask them for those 3 items. And what were the 3 items again? Cheating if you're writing because you see them. There you go, exactly. Right, you can do that. They can't. Once it's out of their working memory, they have no ability to take that information from short-term memory into long-term memory. Right? 2 very, very famous movies, very, very different movies that have been made involving people who have damage to their hippocampus. What are they? Well, okay. Yes, my 13-year-old has trouble remembering things too, but that's typically not hippocampus damage. I think that's just, 50 First Dates, right? A good, hilarious one, right? With Drew Barrymore and Adam Sandler. And then the other one? Memento. Guy Pearce, a guy whose wife, policeman, bad guy breaks into their house, kills his wife, damages him so that he has damage to the hippocampus and cannot convert short-term memory into long-term memory. And he wants to get revenge, kill the guy, but he can't remember anything. So he's got to write down notes and get tattoos on his body and all these types of things to help him to retain this information. Great movie. Movie is actually shown out of sequence to kind of give you that experience of what it would be like. It's an excellent, excellent movie that I highly, highly recommend. Absolutely. So both of those are excellent movies. But again, it gives you that idea of the problems that can have with that. There is a famous individual who's known only as HM as the subject, who has bilateral damage to his hippocampus. He was camping and it started to rain. So he got into his station wagon with, I think, his camping materials, and I think it was propane or the charcoal or something like that. He ended up getting carbon monoxide poisoning. And the carbon monoxide poisoning damaged his hippocampus, and he lost the ability to retain his short-term memory. And that happened close to 40 years ago now, probably that happened. There's been some instructors, one of them at UC Davis, who, especially early on for the first 25 years, basically worked with him every single day. So for 25 years, every single day, he would go into the guy's room and he would have to introduce himself to the guy for the first time. All right, he would work with him, leave for lunch, come back, and have to introduce himself to him again. All right, HM has no ability, has not produced any new memories since that event that has now happened, 40 some years ago now at this point. Now, again, you have to imagine how remarkable that would be, right? Especially as you get to year 10, year 20, or something along those lines. When you wake up and look at yourself in the mirror, are you seeing the person you expect to see? No. Now, obviously, most of us would be freaked out if you looked in the mirror and suddenly you were 20 years older than you thought you were, right? That'd be pretty scary. However, he also has amygdala damage, so his emotions are somewhat muted as well. So he's aware that something happened, that he's had some damage, but he has no sense of time. And a lot of what we've learned about memory, we've learned from people like him as well. Like, for instance, we talk about muscle memory, right? That's something people talk about. Well, that's a real thing, right? Not surprisingly, HM has a tremendous amount of time on his hands. So he plays a lot of video games. So Monday morning, you walk in. Of course, you have to introduce yourself to him again. You show him a video game and ask him, have you ever played this video game before? And he will say, no, I've never played that video game before in my life. You put the video game in, hand him the remote control, and he'll get the high score. The whole time swearing he's never played the video game before in his life. All right, so things like muscle memory, motor memory, the different types of functional memory, a lot of what we've learned are from people like this who have these hippocampus damages. All right. Excellent. Speaking of the hippocampus, there is a paired structure right here known as the mammillary body. Is that on your list?
No.
And why am I talking about it? Because it is on your list. It's just not a telencephalic structure. It is a diencephalic structure. So if you look under your diencephalic structures, and if I'm seeing it correctly, is it number 49? This structure is what is known as the mammillary bodies, and we'll see why it gets its name right. I guess I have to go further than I thought. All right, we'll find it. There it is. Down here deep in the diencephalon are these two enlarged globular enlargements where, again, anatomists, you can see why they call them the mammillary bodies. It looks like little boobies. Right? We can also see it here sticking out, part of that duck shape that we said is the diencephalon. So it is a diencephalic structure. So of course the question is, why are we talking about it here? And we'll see that in a second. But let's talk first about what the mammillary body does. The mammillary body plays an important role in taste and smell aversion. Right? If you eat something that makes you sick, do you necessarily want to eat that thing again? No, and that's kind of what our mammillary body does. The job of it is to be protective in that way, right? And again, the problem is it isn't, this association is very, very strong. My wife, when she was pregnant with our first daughter, we were down in Southern California, and there's this restaurant in Southern California that has this amazing black pepper ranch dressing that they have on their salads that she absolutely loved. So we had gone there for dinner one night, and being pregnant later that evening, she got really, really sick from, the motion sickness, the morning sickness, and things along those lines, which again, as you know, doesn't just happen in the morning. But she threw up a tremendous amount as a result of that. And even though it wasn't the ranch dressing that got her sick, did we ever go back to that restaurant and ever eat that black pepper ranch dressing ever again? No, right? Someone just mentioned how they couldn't eat gorgonzola cheese anymore from being pregnant, or was that just like some kind of drunken stupor thing that you had? Pregnant, there you go, right? Or the example of, I have a friend, and by friend I mean me, who drank way too much tequila one night. They ran out of lemons as chasers and tried to use oranges as chasers, which does not work. And now still this feel of the pulp, the smell of fresh squeezed orange juice still makes me a little queasy, even though that happened 2 days ago. No, I'm just kidding. many years ago, high school, right? And yet still the feel of the smell of fresh orange makes me a little queasy as a result of that. So that is a very, very strong sense of that mammillary body. And if you notice, and this is why we're talking about this right here, this yellow structure are axons. It is a fiber tract that connects the mammillary body, where we talk about this taste and smell aversion, to our hippocampus, where we store our long-term memory. The nice thing about this kind of weird section, this oblique section of the brain, is we can see the entire fornix from this. But notice, with this sagittal section, we can see it here as well. This structure right here is that fornix. So even on a sagittal section, we can see that fornix. And again, fornix is really just a structure that connects the mammillary body to the hypothalamus. So we don't have to worry about a function for the fornix. We just have to make sure we recognize the fornix.
And that fornix you said was axons?
Yep, it is a white matter tract. It is axons. That is correct.
And it connects it to the hippocampus?
Yes, it connects the mammillary body to the hippocampus. So Haley, let's see what you have there. Sure. Well, we'll talk about that. We'll talk about our sense of smell in just a minute, but absolutely, the sense of smell is absolutely an important characteristic associated with memory and things along those lines. And in fact, while they also use essential oils for calming things and things along those lines, they're also trying to use things like essential oils in strong sense to as a treatment for post-traumatic stress disorders. With post-traumatic stress disorders, what has happened is stressful situations have kind of hardwired some of our pathways so that when stimulus A occurs, you have that sense of dread, that traumatic experience associated with it. So what they try to do is they try to decouple that, weaken the strength of that by doing things like using lights or doing things like using scents to kind of disrupt that sensation. One of the reasons that the essential oils and things like that, yeah, exactly, the call is to confuse it, to change the pathway. Here we see it nicely in this illustration, both of these. Underneath the cerebellum is a structure called the olfactory bulb. Here we see the olfactory bulb here. And then that's where cell bodies are located. And then the olfactory tract, which is this bundle of axons, which carries the information from our sense of smell to our brain. If you remember, this is something we've talked about before. I don't, if we go back to this picture. Remember way back in the day when we were studying the bones, we mentioned how this structure right here is the olfactory bulb. And this olfactory bulb is where the axons come off that give us our sense of smell, our olfactory nerves. I told the story about my sister's boyfriend who fell down the flight of stairs and ripped his olfactory bulb off of his cribriform plate, and he lost his sense of smell. Well, as we can see from this illustration, our sense of smell goes straight back to our limbic system, our emotions, our memories. So sense of smell is the one sense that is most closely associated with emotions and with memories. All right. So again, you, like me, stare at those pictures of that long lost love of yours. You may listen to that last phone recording that he put on your phone that you haven't bothered to erase yet. But when I'm really feeling sad and lonely, I get the t-shirt out that he left for me, and I open it out of that Ziploc bag, open it up, and just take a sniff of him, and all those emotions and all those feelings all come back to me, right? Or maybe it's the smell of that baked apple pie that reminds you of that summer you spent at grandma's house, swinging on the tire swing, swimming in the pond, all those types of things. Our sense of smell is the one that is most closely associated with our emotions and with our memories, because it's the only one that goes straight to the limbic system. comes straight into the limbic system. So it's why we have that strong association with smell for memory, for emotions, and that's why the mammary body is so important as well, for protection as well. All right.
Clarify, the olfactory bulb is like the ability to smell, and then the tract takes it back to interpret it?
Great question. No, the olfactory bulb is where the cell bodies are located. And then the tract is the axons carrying the information back. The olfactory bulb actually gets its information from the olfactory nerves. We'll talk about that when we get to the nerves. So what we have here is basically a ganglion or a nuclei, the olfactory bulb, that gets the information and then sends it down the axons to the limbic system. So both are structures helping to carry information into our limbic system about our sense of smell. You don't have to worry about distinguishing different functions for the tract and for the bulb. Just be able to recognize them and know that both play a role in our olfactory pathway. All right. All right, 3 more structures. Let's talk about the largest structure found in the limbic system, and that is this big white matter comma-shaped structure called the corpus callosum. The corpus callosum is our largest commissural tract. Remember, commissural tract are axons that go back and forth between the 2 hemispheres. We see that corpus callosum really nicely in this image here, but the other place where you really get a good sense for what and it's really great there as well. But the other place where you really see it get a good sense of what it does is right here. Notice this right here is our corpus callosum. Okay, I did that a little big. This right here is our corpus callosum. A big, huge bundle of axons carrying information from the right side of our brain to the left side of the brain. It is the largest commissural structure. And as we talked about, is it important to coordinate what happens from the left side of your brain to the right side of your brain? Yeah, absolutely, that is important. So if this were to accidentally, say, be cut, that would be a devastating injury that would really impair someone's ability to function normally, right? What do you think? Do you think you could function normally if someone cut your corpus callosum? I got 2 no's. I got a sometimes, but I like Adina's answer the best. Yeah. Shockingly, it actually is very little deficit that we would see. Now, of course, the question becomes, why would you cut someone's corpus callosum just for the heck of it? And the answer, of course, is no. However, there is a very severe condition called epilepsy. What does epilepsy involve?
Seizures?
Seizures. Seizures basically occur because what happens with epilepsy, a region of the brain, and let's just for argument's sake say it's this region right here, produces a big, huge, massive neural signal, what they call this neural storm. And this neural storm basically spreads across the entire hemisphere of the brain, crosses the corpus callosum, and spreads all over the other side of the brain. And you have these big, huge neural storms that cause these massive grand mal seizures. These are the ones where the person is flailing on the ground, where you have to put a wooden spoon in their mouth so they don't bite their tongue off, right? Incredibly debilitating condition for an individual to have. And it's very hard for them to live a normal life. Not only do they have a hard time just sitting in the classroom, right? They have difficulty, they can't drive. There's all sorts of other limitations of them because of this huge devastating condition. But typically these neural storms just occur in one consistent location. So what they will do in these individuals is they will go and they will make an incision through the corpus callosum, producing what they call a split brain. Now, when that massive wave occurs and spreads across the hemisphere, it stays limited to just that one hemisphere and isn't able to spread efficiently to the other side and cause damage. And it dramatically reduces the severity of the neural storm, can dramatically decrease the severity of the seizures. And in some cases, these people can lead relatively normal lives. Now, again, when this was first discussed and talked about, this was thought to be the lesser of two evils, because of course we thought that cutting someone's corpus callosum would really impair them functionally. But it turns out that the nice thing about the world is fairly consistent. If I see an object in front of me, that object is in front of me. And so as long as my right side can see it and my left side can see it, it's not so important that the two sides of my brain actually communicate with each other to be able to do that. Is there some loss of coordination, other things like that? Sure. But what's remarkable is that these people can behave in many instances almost completely normally. And it's impossible to know they have this disorder. Now, they have figured some ways out to tease out this deficiency. What'll happen is they will look in a special viewfinder. And in that special viewfinder, it will show them an image of a person's face. And what'll happen is the left side of the face will be male and the right side of the face will be female. So when they look through the image, what happens is they'll tell them, all right, write down what you see and say what you see. And they will say male and write down female, and they won't realize that there is a discrepancy there. But notice it takes a really specific, unique kind of situation like that to tease out that difference. Shockingly, these people are can lead relatively normal lives with that. All right. Now notice one other thing, actually two other things that are here as well. There are spaces in the brain. While there's all these nooks and crannies, there are also some fluid-filled spaces in the brain. There happen to be 4 of those, and we'll talk about them a little bit later. But here we can see 2 of them, and these 2 spaces are known as the lateral ventricles. These contain fluid, and what kind of fluid do you think they contain?
Cerebrospinal fluid.
And remind me again, what are the glial cells that are responsible for helping to produce and maintain the cerebrospinal fluid?
Ependymal.
Excellent. Excellent. So these ventricles are lined with ependymal cells. They contain cerebrospinal fluid. And like I said, there's 4 of them, including 2 lateral ones. And notice there are 2 lateral ones because they don't touch. They don't meet in the middle. Notice that just inferior to the corpus callosum is a thin membrane that separates the 2 lateral ventricles from each other. This dividing structure is called the septum pellucidum. Right? Just like we have a dividing structure in the middle of our nose, the septum, here in the brain, we have a midline septum as well, known as the septum pellucidum. It's super easy to see here in our coronal section, but notice also if we look at our sagittal section, remember here is our corpus callosum. Remember, here is our fornix. And notice this thin membrane right here in between, that is the septum pellucidum. So notice right now where my pen is, my pen would be in the left lateral ventricle. And if I poked through, I would be in the right lateral ventricle. So if we go back to the previous picture, basically that image, my marker was sitting right here. And if I poked through, I would be in the ventricle on the other side. Does that make sense? And again, it is super easy to find here, right on the midline. Septum's on the midline. That makes that super easy. But even here, like I said, between the corpus callosum and the fornix, that right there is our septum pellucid.
So would you ask this by saying like identify the space, and that would be the lateral ventricle, and then the structure would be the septum pellucum?
Exactly. I could put an arrow here like this, and you're absolutely correct. I could ask those two questions. Identify the space, identify the structure. Same arrow, different keyword, so different answer. You're absolutely correct. If I ask for the space, it is going to be the lateral ventricle. If I ask for the structure, it is the septum pellucidum. Now notice it's a little bit easier if I go here, because here I can now use different arrows. Identify that space, identify that structure, right? So this way it's easier to tease them apart. But technically, could I ask it from the other view? Yes.
Do we need to identify left and right?
Should you know left and right at this point? Yes. Would I mark you wrong if you didn't put left lateral ventricle? No, I would be fine with just lateral ventricle. At this point, I think I can assume that you guys know the difference between right and left. And if not, then you have other problems in life. All right, one more. Here in our limbic system are these three nuclei. Now, these three nuclei each have their own name. But like some of our muscle groups, remember in a muscle group, I just needed you to know the muscle group name and not the name of the individual muscles. That is the same thing true here. These things collectively are known as the basal ganglia. Now, there's a huge problem with that name. Remember, a ganglia is a collection of cell bodies outside the central nervous system. This is in the central nervous system. So since the beginning of time when they first started calling this something, they called it the basal ganglia, which is a huge misnomer. They are getting better about it. More and more you will see in the literature that it is now referred to as the basal nuclei. But because basal ganglia is still used so much in the literature, I will technically accept both terms for it. So you may call it the basal ganglia or the basal nuclei, and I will accept either of those names. So call it whichever one you like, but most importantly, let's talk about what it does. What our basal ganglia does for us is, and again, here's the fancy term for it. Let me give you this: subconscious control of skeletal muscle. Excellent. So now that we've written the fancy term that we can call grandma and say, hey, guess what, grandma? The basal nuclei gives us subconscious control of skeletal muscle. Let's talk about what that actually means. The basal nuclei does not initiate or terminate actions. What it does is it maintains a learned action. Oops, where did that go? Essentially, it's your autopilot. All right? How many people need to think about what they're doing while they're walking these days? Not too many of you. So you eat a sandwich and you text, you talk to the person next to you, do your makeup, all sorts of number of things that you do while you're walking down the street, right? But what about that 1-year-old? Can they text and walk at the same time? No, it requires every single ounce of their focus on thinking about what it is that they are doing. Or let's take it to the current example that I'm dealing with. I have a 16-year-old who has her learner's permit and is driving the car. When she's in the car And we just went on the highway for the first time this past weekend with her, right? 100% of her focus is on driving, paying attention, right? Thinking about what she's doing, looking at her environment. Is that how things are when you're driving? No. Again, when you're driving now, you're talking, you're texting, you're eating that sandwich, you're doing the makeup, you're doing all those other things. However, that's a problem with an autopilot, because while you're, sure, yeah, we know you, Haley. You're one of those young ones who can hear the sound. I guarantee you're texting while you're driving. All right, or tweeting, or I'm on Grindr while I'm driving, whatever it is. Right, we're all doing those kind of things. We're all the same. When you're on there and doing those things, your autopilot kicks in, and suddenly you're halfway to work, and you were supposed to be going to the store to get milk. Right, that's the one problem with that autopilot, is that autopilot kicks in, and you end up going the wrong way. Right. See, there you go. That is very responsible, Haley. And you're probably, especially with only 12 of us here right now, I guarantee you're the only one in the class who does that. But you can tell how guilty Laura looks there. She definitely tweets while she's driving. See, there you go. Well, that's a good thing, right? And again, you're doing that. My wife, a couple winters ago for Christmas, we went and visited her grandmother, and her grandmother tried to teach her how to knit, right? Again, my wife, every focus was on every knit, every purl while she's doing that. Is that how grandma is? No, grandma's going away, having a conversation, watching TV, baking a pie, doing all those things while she is knitting. Again, she has that learned behavior, and your autopilot maintains that learned behavior, keeps that learned behavior going. Right? Yeah, it's fun watching movies if you can knit. Once that knitting is a learned behavior, then yeah, but you got the young ears though, so that's all that matters. You got the young ears, so there you go. So actually, knitting is coming back. It's like it's the new hip thing to do, right? All right. So that is these three things together. They form that autopilot for that learned behavior. Yeah, knitting or baking, right? Those are the two things that most people have picked up while in quarantine. All right. And that, I think, is everything on your list for the limbic system. Am I correct?
Fornix?
We did the fornix. Remember, the fornix is that white matter. Notice, remember, you see it's white matter. It's axons going from the mammillary body to the hippocampus. So here we see it in its entirety. And here we see it on the sagittal section. So I think that is everything for that. Now, we've hinted at the flow of the cerebral spinal fluid, but we will, how are we on time? We're doing good. Give me a second. We're doing good. So that has gotten us through most of our telencephalon. We do need to talk a little bit about the flow of our cerebral spinal fluid, but I actually want to do that at the end after we do all of our cephalons. So with that, we are done with everything that is in the telencephalon. And I think that that is a good stopping point. I know it's a little bit early, but I think we've caught up a fair amount, and I think we're going to do good and still get to the cranial nerves on Monday. So I think we're doing good on time. So this is good. So let's go ahead and finish a little bit early, get an early start to your Thanksgiving holiday. So I think this is a good stopping point, unless there are any questions you guys have. No questions? Oh, there you go. Exactly. Happy Thanksgiving all then. Have a good, safe Thanksgiving. Be responsible this evening. Do a lot of, not just reading, but studying. No fun reading, all studying. Be safe, wear your mask, stay at home. Have a happy, safe Thanksgiving. Have a safe, happy Black Friday. And I will see you guys on Monday when we come back. All right. And remember, have your cranial nerve handout for Monday, because we're going to hit that. That's going to be a big part of this next exam. So make sure you're doing that as well. All right. Take care, everyone. Bye.